Let's look at ways to use phage transduction to characterize suppressing mutations. So let's look at the example on the bottom first. So we started with a strain, and that's shown here, that had a MET minus phenotype, and we now have MET plus. We're going to do a phage transduction to determine if that's due to a reversion or a second site mutation. So we're going to grow phage on this strain, and remember that the phage, some phage will package some of the chromosome, and we're hoping that these regions get picked up by some of the phage. Then we're going to take that phage and we're going to infect a new strain, a new cell, and this is an E. coli that is ARG minus MET plus. Arginine, it requires arginine for growth, but it's MET plus. Now we're going to demand that they grow on medium without arginine. So the wild type copy of the gene has to come from one of the particles that was packaged in the phage head. And in this case, we can see that we're going to select ARG plus and ask how many of them are also MET plus. And in a true reversion, 100% will be MET plus, and that's because if you recombine here, you're bringing in the wild type copy, which is, we've already established, is a true reversion from the strain that the phage were grown on, or if you recombine here, this parent strain was already MET plus. So 100% tells you that it was a true reversion. Now let's look at a situation where there is suppression, and here we have our same mutation that we started with, so they're MET minus, they're ARG plus, but we think we have a suppressing mutation. We grow phage, and again we're going into an ARG minus MET plus strain. Everything will have to be ARG plus, and we'll ask how many of them will be MET plus. In the picture shown up here, we're looking at a situation where the DNA brought in on the phage head gave us that ARG plus, because that's what we're demanding, but it did not recombine to bring in the MET mutation that was on that original strain. So these end up getting the ARG gene from strain 1, but the MET wild type gene is actually from this second strain, strain 2. And 86% are going to be of this genotype. And part of that is because we know the distance on the chromosome between arginine and methionine. But now let's look at another situation. In this case, we get a recombination event that, of course, gives us the arginine gene from strain 1. But we will get some recombination events that actually allow crossover of that methionine mutation that was on strain 1. So these will be ARG plus MET minus. And that tells us that the original methionine mutation was still present in strain 1, and that the phenotype of that strain had to be due to a suppressing mutation. I want to point out one thing that might be a little confusing when we look at the top here, infection of ARG minus, MET minus. These are both this suppressor strain and this MET plus are the MET plus phenotype. So I think this is just to indicate that the original phenotype was MET minus. There are other things that you need to do to really analyze a mutant. So let's say you're starting with your mutant, you have a phenotype. one thing you need to do is prove that there's a direct relationship between the mutant and the phenotype by complementing the mutant. That means you add back the normal gene to the wild type version of the gene to that mutant and restore a wild type phenotype. You might need to determine if the mutation acts in cis or trans, which we'll talk about. You would test for allele specificity, particularly important if you're looking at a pathway, and finally determine if a given mutation is dominant or recessive. Let's start with a simple example of genetic complementation. So you've got a mutant that is a thymine auxotroph. In other words, these cells will not grow unless you add thymine to the medium. Now you know from other studies that there are two important genes, Thi-A and Thi-B. So the very simplest experiment you could do here, given that you have all this information, and by the way I'm simplifying this, you could use PCR to amplify each of those genes, clone them into a plasmid, so you've got A here and B here, and now add both of those plasmids to your mutant. Separately, of course, by electroporation, or get these in using one of the methods that we talked about earlier. And in this case, we would see that when you plate on just regular medium with no thymine, you got colonies here, but in this case you did not get any colonies. You would conclude that your mutation was most likely in the Thi-A gene. Now let's say you've got that same Thi mutant and you don't have access to a PCR machine and you don't have any other easy way to locate this gene, and you want to get the entire Thi-A gene. Here's a trick that is a very powerful way to get the gene of interest out of a cell using a library. And we've talked about libraries earlier, so you know how to make a library. What you would do is take your Thi-A mutant and you would transform with a library, and you would then plate those transformants on medium without any thymine, and you would get very few, but you only need a couple of colonies that can now grow. Remember, you would probably on your library you would have some antibiotic resistance, maybe ampicillin resistance, so these would be plates that are contain ampicillin and they lack thymine, and your two colonies here would probably contain the gene that you're looking for on a plasmid. And sure enough, when you study those, you find that here's your plasmid. It's got the AMP gene on it here. It's got an origin, of course, and it's got, in this case, the complete Thi-A gene, because it could complement, you know, it has the gene and it has its regulatory elements upstream, because it was able to express that protein, thymidylate synthase, from the plasmid, and that complemented this mutant. I'm going to use our old favorite lac operon to remind you about the differences between cis and trans as genetic terms. So remember, we've got, this is just looking at the regulatory region of the lac operon. So here's lac Z, and we've got a promoter in this region, E. coli is, in the lac-Z operon in particular, has an operator region, and the operator region is a part of the chromosome to which a repressor protein will bind. Now notice this repressor is going to bind right to this operator region in front of RNA polymerase, so it blocks RNA polymerase from moving forward and making mRNA. Now this repressor is actually encoded by the lac-I gene. It's called the lac-I protein, the repressor, and it is always being made. Now you'll remember that that repressor comes off the DNA when allolactose is bound to it, because allolactose basically inactivates that repressor and prevents it from binding up here to that operator region. There are other components that we can use in it. Instead of allolactose, IPTG is one, called a gratuitous inducer. But the normal molecule is allolactose, which is produced when cells are on lactose. So even though the lac-I is still being transcribed and translated, the repressor is inactive, and RNA polymerase can now move forward and make mRNA from this region to make the three proteins, beta-galactosidase, the permease, and the transacetylase, that allow cells to grow on lactose and give us blue colonies on xgal. Well, let's look at how this system can be used to understand some of these genetic elements we're talking about. So here's a cell, and we're going to just focus on a small bit of the operon. We've got lac-Z, we've got the lac operator, and we've got a mutation in lac-I. So without any lac-I, that means there's no repressor. We don't need anything to come off the DNA because it's not bound. And that means that the polymerase will be able to transcribe this region, and translation will occur as normal. So the color on xgal will be blue because there is no repression. Now let's add lac-I and wild-type lac-I back on a plasmid. And that's shown here. And let's think about what that situation is going to be. Now we can make the lac-I protein, and it can bind to that operator. And what's that going to do? It's going to block transcription. So color on xgal, and we're assuming that we haven't added any inducer here, is going to be white. So that example indicates that plasmid-borne lac-I can complement that lac-I mutation. Well, let's look at a different example. Now we've got a mutation in the lac operator, and we've added the lac operator region on a plasmid. Now what we find is that that plasmid-borne lac-O does not complement, even though it's wild type, it does not complement that mutation that's on the chromosome. So is that cis or trans? Well, let's look at these. The situation up here where we've got lac-I being able to complement tells us that lac-I can act in trans. It can be produced someplace else and have an effect elsewhere on the chromosome. So even though this is plasmid-borne, it still functions because that lac-I is binding to the lac operator region here. Lac-O, however, is a cis element, because even though we've added the wild-type lac-O here, it has no effect on what happens with the LAC-O region or the other flanking genes on the chromosome. If you remember when we were talking earlier about the experiments of Beadle and Tatum, I mentioned that the histidine biogenesis pathway is fairly complicated. There are many genes. So allelic testing is done if one is trying to compare two different mutants to see if they have mutations in the same gene or different genes in a pathway. So let's look at mutant 1, and it's a His minus mutant, and the first example here that we'll talk about is a situation where we've got two different His mutants, mutant 1 and mutant 2, and at the outset we don't know where the mutations arise in these pathways. We simply know that they're both His minus. For example, if we've got in one strain a mutation in His A, but otherwise it's wild type for His B, and our second strain has a mutation in His B, but is otherwise wild type for His A, if we take the genetic material from strain 2 and, for example, put it on a plasmid and add it to strain 1, then we should be able to get a His plus phenotype, because we have got the potential to make His B and His A, and so we've restored the pathway. Well, let's look at an example of a different mutant, also a His auxotroph, and we're again comparing two strains. Strain 1 has a mutation in His A1. Strain 2, we call it His A2, but that means it's a different mutation in the His A gene. The mutation is here. So in both cases, we will not make any His A protein from either of these alleles. However, we'll make His B in both cases. Now you can, I think, pretty easily predict what is going to happen when we try to combine those. If we took this region of the genome from mutant 2 and put it on a plasmid and added it back to strain 1, we would make a lot of His B, but we would still not make any His A protein that was functional, so we would end up with a phenotype that is His minus. Well, let's look at one final genetic test that's important to do. And again, here's our example looking at the lac operon. And we've got lac-ZYA, lac operator, and lac-I is being made under normal circumstances to bind to the operator. So if there's no inducer, either no lactose, our inducer is lactose, or IPTG, for example, we don't make any beta-galactosidase because the lac-I protein is bound to that operator blocking transcription. If we add either one of these, lactose or IPTG, we've already examined this. Lac-I will bind to those inducers. It will no longer be able to bind to the operator, and beta-galactosidase will be made. Now we're going to add a plasmid to this strain, and this plasmid contains a lac-I mutation, and it's called lac-I 66. And when we do the experiment again now, we find if we don't add inducer, no beta-gal, so that's just like in the wild type strain, the normal situation. However, now when we add inducer, we still don't get beta-gal being made. So what is going on here? Well, this is an example of something called dominance. Because this mutated lac-I makes a protein product here's my LAC-I, and it is able to be dominant over the LAC-I because it's going to bind to this operator region and hold on even when inducer is made. It doesn't matter if there's a wild type copy of LAC-I there. It is ineffectual compared to this inducer or this LAC-I, which can no longer bind to the inducer. In this last section, I want to go over a few more molecular tools. I'm not going to talk about transcriptional and translational fusions. We've talked about those before using reporters such as LAC-Z or GFP, for example. I'll mention a few more techniques that involve using GFP and its other varieties such as YFP, yellow fluorescent protein, or M-cherry, which is red. And in the cell you can actually make multiple fusions using these different fluorescent proteins so that you can watch multiple proteins in real time to see what they're doing in the cell and whether they interact. I'll talk about an epitope tag, and the example I'll give is the his tag. We'll talk about the two hybrid approach, and finally I'll mention mass spec. There is another technique that I won't have time to talk about today that uses 2D gel electrophoresis, in which you analyze proteins that have been labeled differentially under two different circumstances, and that's a pretty useful approach. This is a figure from your book that might be helpful to organize your thoughts on how all of these processes are related. So for example, we've just been talking about fluorescent tags to localize proteins. We've talked about suppression. We'll talk in a couple of lectures coming up about operons, regulons, etc. We've talked about RT-PCR microarrays and looking for small RNAs using RNA-seq, for example. We'll talk today about the two hybrid techniques, and of course all of this relates to looking at functional annotation, taking DNA sequence, looking at the predicted mRNA, and finally looking at the predicted protein structures. Well, let's say you've just taken a new job in a lab that studies a pathogen, and you're looking at virulence factors, genes that encode proteins that affect the host. And you've been given the task of looking at a protein called VirX. And your boss wants you to determine if VirX has the enzymatic activity that's been predicted for that, and also might want you to determine where VirX is located in the cell during an infection. So here's what you're going to do. Instead of trying to purify VirX the old-fashioned way and get antibody, which would take many months, you decide to clone the VirX gene. So you maybe use PCR to amplify it, and you're going to clone it into this vector. So this is a PET vector. If you remember, that stands for expression technology. And here are some of the features. It has an origin, it has ampicillin resistance, it has the LAC-I gene, which we'll talk about in a minute, and then it has this important region up here, and let's look at that in more detail. So shown below, we're going to take our VirX DNA that we've cloned, amplified by PCR, I should say, and we added BAMH1 sites to the end of it, and we're going to clone it in the proper orientation into this BAMH1 site. So here's VirX, right there. Let's look at the other features of this region and what's going to happen. Here's a promoter. Here's the LAC operator, which LAC-I will bind to, allowing us to regulate when we make our construct that I'm going to talk about. So we've got mRNA, Here's a ribosome binding site. Here's an ATG. All of the elements that we've been talking about are present. And there's something else. There's a bunch of codons that will encode for histidine. 6 histidines in a row. There's a thrombin site, which will become important later. And now when you, in E. coli, add an inducer to remove LAC-I, we'll make mRNA, we'll translate this region, and we will get a His6 Vir-B protein. Now here's what that allows us to do. This is pretty nifty. It turns out that when you have a configuration of 6 histidines, that allows you to use a nickel resin or some kind of a matrix such as a magnetic particle, that's what's shown here, bound to nickel, and any protein that has a His6 configuration will bind to that. And again, it's these 6 histidines in a row. Sometimes it's a His8. But now you've got your cells that were expressing this fusion protein. You've lysed them, and you've got it bound to this nickel resin. Oh, sorry, here's the nickel resin down here. And here's your favorite protein. And the His region is shown in the little purple circle. And now you can take away everything that you don't want. You've got your protein bound to the nickel resin, and in the end you elute it, and here is your Vir-X, your His6 Vir-X. You can use this thrombin cleavage site to actually get rid of the His6 if you want to have final Vir-X alone. You can also use this fusion protein in the cells because you can get antibody, commercially available antibody, that will recognize that His6 region. So you can now basically in a very short period of time use a technique that allows you to track where Vir-X is located in the cell. So it's a pretty nifty technique that incorporates a lot of molecular tools so that you can both purify a protein and study its enzymatic activity if that's your goal, or look at the cellular location and expression pattern. Sometimes we use reverse genetics to learn more about our organism of interest, and we could use proteomics and mass spec analysis to understand and identify a gene of interest. So for example, we would start with a sample and fractionate. For example, these are not bacterial cells, but we could take our cells and separate the cytoplasmic portion from the membrane portion. And then we would run each of those samples on an SDS phage gel, that's polyacrylamide gel electrophoresis under denaturing conditions, and we would set this up in such a way that we had a pretty clean band that we could actually cut out of the gel. Those proteins would be subjected to trypsin digestion, and we would get peptides. Those peptides get used, separated chromatographically, and using electron spray ionization, we can separate all of those different peptides. That can then be followed by mass spec or tandem mass spec to give us kind of a signature for all of the different peptides that were generated from the protein that was excised from that gel up on the top. And then you can use that information, and there are a number of database systems that are and software, that you can basically input all of these peptides, and it will tell you what gene they came from in the database. So this is reverse genetics, well, part of reverse genetics, where you start with the protein and work your way back to find the gene of interest, and then you can make manipulations of that gene. I want to remind you about the basic types of fusions A transcriptional fusion can be very useful if you're trying to study what is happening with a given promoter, and that in that case we're putting LAC-Z or some other reporter gene in an operon so that we can monitor that promoter. We're not making fusion proteins though. A translational fusion allows us to make actually a fusion protein. Recall that these fusion proteins were useful in studying secretion and would also be useful if you're trying to determine the cellular location of, for example, ORF-A product, and in that case it would probably be very easy to fuse it to GFP so that using fluorescence you could see in the microscope where the normal position for the ORF-A protein was. Here's a cartoon showing you how you might engineer one of those fluorescent fusion proteins. Here's our starting plasmid. It's got GFP in it, it's got a promoter, and it's got a restriction site and a terminator. So we're going to put our favorite gene, XXX, in here. In this case, we're adding IPTG, so this must be the LAC regulatory region, and we produce a fusion protein in which GFP is at the amino terminal end. Of course, GFP could also be put at the C-terminal end of your gene so that your fusion protein has the configuration XXX GFP. Let's look at a genetic tool that allows you to look at very specific protein-protein interactions. This was a technique that was developed by Stan Fields and one of his students. So let's look at the background part of this first. So they were working with yeast, Saccharomyces cerevisiae, and they knew about a protein called the GAL4 transcriptional activator, and it typically binds to an upstream activating sequence in DNA, and it's required to activate RNA polymerase for transcription. Now here's the interesting thing about GAL4. It has two important regions, a binding domain shown in green and an activation domain. And they decided to take advantage of those two regions of the protein to see if they couldn't develop a tool for looking at protein-protein interactions with other proteins. So here's what they did. They separated the GAL4 gene into two regions. They took the part of the gene that would encode the binding domain and the part of the gene that would encode the activation domain, and they put them on two separate plasmids. So now we're going to look first at the binding domain. And in this case, they took the binding domain and they made a fusion protein with a gene encoding what they call BAIT. So this was the gene for a protein that they were looking for interacting partners with. They would make a fusion protein, of course. It would have the binding domain, the GAL4 binding domain, fused to the BAIT protein. And the GAL4 binding domain can still bind to this upstream activating sequence, but we see no activation of transcription because the GAL4 activation domain is not present, and that's what RNA polymerase needs. By the way, this should be LAC-Z, lowercase LAC-Z in this picture. Well, the next thing they did is they took the other part of GAL4, the activation domain, and they fused it to a different protein. So in this case, here's the activation domain, and they joined it now to the gene for what they're calling prey, and they think that prey might interact with BAIT. So in this case, we also see that there's an unproductive stimulation of transcription. There's nothing that's going to happen here because there's no binding domain. And this is our control to show that if we fuse prey to activation domain, we don't create some unusual binding to the upstream activating sequence. Well, finally what they did is they put both plasmids into the same cell. So now we've got our binding domain bait, so that was the first plasmid that was made, and we have the prey fused to the activation domain plasmid 2. Now when we express both of those, we have our construct here, and if the bait and the prey can interact, that is sufficient to bring the activation domain, or the binding domain and the activation domain, into juxtaposition. The binding domain can still bind the UAS. Now the activation domain is present and it can interact with RNA polymerase, and we can stimulate transcription and we will get blue colonies on X-gal. Very clever technique to prove that two proteins interact. The bait and the prey had to interact in order for this stimulation of transcription. They went further and they showed that you could take your bait construct and you could actually make a library in the other plasmid. So let's say we're looking randomly on the chromosome for a gene encoding a protein that will interact with bait. So now they make a library in the activation domain plasmid. So we've got all sorts of pieces of DNA in here, many copies of this activation domain plasmid. We now mix those in cells with the construct that contains our bait, and we look for blue colonies. And those blues end up helping us identify the gene that was productive in the activation domain to give us an interaction, and that will help us identify the gene encoding a protein that interacts with bait. Many proteins in a cell interact with different types of protein partners, and it's often important to know which proteins are pairing up at specific times during the life cycle or during a particular process. But until recently, it's been kind of challenging to do that. But here's a very clever technique that was developed, and it's sort of a hybrid of the techniques that we've been talking about. It's called bimolecular fluorescence complementation. Bimolecular fluorescence complementation. And it takes advantage of the fact that these fluorescent proteins that were originally identified from jellyfish out in the ocean come in different forms. So we've got GFP and red fluorescent protein, and yet there are common components to that. So what they were able to do is, in testing, for example, this protein in the middle, they wanted to determine when it interacted with partner A, this striped barrel, or protein B, the vertical striped barrel. And they could fuse part of the fluorescent protein to our test protein here in the middle, the red barrel. And then they fused the other part of the fluorescent protein, the green part of it, so that if there was a productive interaction between our test protein and protein A, we would get green fluorescence. And that's shown in the figure below. In contrast, when our test protein interacted with protein B, we would get this interaction, and now we would bring these two components together, the top components, and in this case we've got the part of the protein, the fluorescent protein that is joined to protein B, is a red version of the fluorescent protein, and now we would get a red fluorescence over here, and that's shown in the picture below. So it really allows you in real time to see how fast these interactions are changing and when they're changing in the real life of the cell.